Okay, so we left off talking about action potentials, which remember is how the nerve signal is actually sent down the axon, right? So an action potential is just the name of the nerve signal. And it has to do with the charge, the potential across the membrane. So normally at resting potential, we said a neuron at rest has the sodium-potassium pump acting. So it's got 3 sodiums going out, 2 potassiums coming in, so that the inside of the cell is very negative compared to the outside at about negative 70 millivolts. That's what we say is resting potential. A resting neuron has that potential. What happens is during an action potential, during a signal, when a signal is sent, that resting potential changes. So it moves from negative 70 millivolts, and that happens through the action of the afferent signals, which are going to send their signal by either opening up channels, allowing positives to come in, or closing channels, making positives not be able to come in. So it's either going to make it more negative, if it's a negative signal, or more positive, if it's an excitatory signal. So an inhibitory signal is going to come down, an excitatory signal will come up, and that'll keep going until we hit threshold. Once you hit threshold, which is at negative 55 millivolts, so if the difference in charge between the outside and the inside gets up to negative 55 millivolts, that's the magic point at which an action potential will be sent. It sets off a whole chain of changes that then travels down the axon and sends a signal ultimately to the axon terminal and the next cell. So let's see what happens once we hit threshold. So we get inhibitory signals, we get excitatory signals. Eventually, we get enough excitatory signals that we're up to threshold, negative 55 millivolts. What that does is, remember we talked about voltage-gated channels? Well, at that negative 55 millivolts is when sodium channels in the cell open up. So sodium, remember, has been pumped out of the cell. So there's lots of sodium outside of the cell, lots of sodium on the outside of the cell, and like no sodium on the inside of the cell. So once they open, the sodium rushes in. Well, what's that going to do to the membrane potential? The difference in charge. That's going to have a ton of positives coming into the cell, which is going to make the inside of the cell become much more positive. So on the graph, we see our potential, our membrane potential shoots all the way up. It hits 0 where there's no difference, and then the sodium just keeps coming in, so it gets more and more positive. Makes it up almost to positive 40. That's the top part of our action potential. That's where the most positive point that we reach. And at that point, the sodium channels close. Remember, they're voltage-gated channels, so they're controlled by electricity and the charge. At this point, the sodium pumps close down, so no more sodium can come into the cell, which is why it doesn't become any more positive. Now, on the way up, another set of voltage-gated channels opened, and those were the potassium channels. Now, the potassium, remember, there's a ton of potassium on the inside of the cell because the sodium-potassium pump has been pumping potassium in. And so when those open up, the potassiums are going to leave the cell. That's going to have the effect of making the cell become more negative. Now the positive charge is leaving. So now the potassium is leaving. The sodium can't come in anymore because they closed down. So now the membrane potential drops. It's becoming more and more negative because the positives are leaving the cell, which is getting us to return to more of our resting level. This is what we call repolarization. We're sort of setting our cell back up to the appropriate difference in charge, right? This part was called depolarization, where we're actually getting rid of the normal difference in charge of the cell. So we depolarize on the way up, we repolarize on the way back. And this is done by the sodium and potassium essentially switching places. The sodium has rushed into the cell, and the potassium has gone out of the cell. So they've swapped. They're now on opposite sides. As we repolarize, there's this important, actually, undershoot here, where the negative charge of the cell becomes less than resting potential. And that's sort of important, and we'll see in action potential propagation, because it means that the action potential can only move in one direction, because it takes time for us to get back to normal. And so it's going to keep that action potential only moving down the axon. Once we reach that negative 55 millivolts again, the potassium channels close, so now potassium can't go out of the cell. The sodium channels are reset back to their original position, and then the sodium-potassium pump takes back over and gets everything back to normal. It's going to get that sodium back inside or back outside and that potassium back inside. It's going to make everything sort of reset back to normal, back to resting, and then the nerve is ready to move on again. That undershoot is responsible for what's called a refractory period, a period during which the nerve is not capable of sending another signal until the sodium and potassium are put back where they belong. Because remember, our sodium came all into our cell and our potassium went all out of our cell. Once the sodium-potassium pump gets everything set back to normal, then the neuron can send another signal. But it has to overcome that refractory period, that sort of resting period, as the sodium-potassium pump fixes everything, gets the sodium out of the cell, brings the potassium back in, sets everything back to normal. Then we can have another signal sent. And so that's how an action potential works. It's all about these ion channels opening up, allowing the sodium and potassium to switch places, and then getting everything reset back to normal. So you can sort of see a general trend in this graph over here. First, you start out at resting potential. You're getting lots of signals in the cell body that are changing that resting potential. Some are making it more negative, some are making it more positive, negative, positive, negative, positive. But once it hits threshold, then you go through depolarization. You hit the peak up at about, in this graph it's saying positive 30. It's between positive 30 and positive 40, depending on the cell. Then you go through repolarization. So depolarization was when the sodium was coming in. Repolarization is where the potassium is going out. That repolarization actually happens too much. It goes below resting potential, causing hyperpolarization. And then the sodium and potassium pump resets everything back to normal. And this happens all along the axon through what's called action potential propagation, where the action potential actually spreads down the entire axon. And that happens because once the sodium is in the first part, it actually diffuses down the axon, which will cause the inside over here to become more positive. And once that hits threshold, then that action potential happens in that portion. And then the sodium spreads some more, and that action potential happens in the next section. So it hops down the neuron. What's important is that undershoot. Remember how we had the hyperpolarization that had to be reset in the action potential? Well, that hyperpolarization keeps the signal from being able to go backwards, because the sodium will diffuse in both directions. But because of the hyperpolarization and the need to reset everything back here, the signal can't actually move in that direction. That way the signal can only move in one direction, only down the axon, until it reaches the axon terminals, where then it sends out those neurotransmitters to their effector cell. And that's how it jumps down. So the sodium is going to keep traveling down, and then it would travel down here, and then we would have another action potential event here, and that goes all the way down. And this happens really, really quickly, milliseconds, right? The response time of a nerve. The myelin sheath helps to make it more efficient because it actually blocks up those sodium and potassium channels where the sheath is. And so your action potential only has to move from the node of Ranvier to the node of Ranvier. So you don't have to go down the entire length of the axon. You only have to go down, you have to shoot off an action potential here, then you have to shoot off an action potential here. But all of this is covered by myelin sheath. So it reduces the amount of area that has to go through an action potential and makes the transmission of the signal much more efficient. Now, I know that that's a lot of information. The action potentials are actually really complicated and hard to understand just from a PowerPoint. So I'm going to link another video, a really well done, professional video that has great graphics and everything. That's going to put all of this information from the last two videos together about how a neuron sends a signal. So you're going to watch that video next, now that you've got sort of the basics down and you understand, hopefully, at least a little bit about how an action potential is sent down a neuron. You're going to see it all put together in a more professionally done video with graphics and animation that should help you to understand it even more. So watch that video next, and ask Mr. Jones any questions if you need some help with this stuff.